A very good morning and good evening to one and all. This is the commercial relationship disclosure for the project that I would be presenting. The study was supported by partnership grant from the DBT Government of India to LV Prasad Eye Institute and Hyderabad Eye Research Foundation. The title of the topic that I'm going to discuss today is Binocular Summation of Cone and Rod Enhanced Specific Signals in Flicker Threshold Task. So before going into the topic, I would be giving a very brief gist about what is this and why are we doing this. So if you see over here in the left-hand side, we have the spatial contrast sensitivity function, and on the right-hand side, we have the temporal contrast sensitivity function. Now, both these functions have been measured monocularly and binocularly, and we can see a clear difference between the monocular and binocular measurements in both the functions, right? Clearly, the binocular sensitivity is better than the monocular sensitivity in both the functions. But in spatial domain, we do know that the binocular advantage is quantified as 41% compared to the monocular visual outcome that we measure. But does that apply to the flicker or the temporal domain as well? The quantified spatial summation, is it comparable to the flicker summation that we see was the question that we had in the first place. Now, taking you all to a very recent meta-analysis by Baker and LIGO in the year 2018, where they have included 65 studies, both from spatial and temporal domain, and have plotted their summation ratios that were measured in all of these studies. For a minute, if we ignore all the spatial studies and focus on few of the temporal studies that were involved in this paper, the summation ratios of the temporal domain tend to have a lot of variation within these results that they have found, and it is clearly not comparable to the spatial domain. So with this meta-analysis and few of the lacunae that we found in the previous study with the temporal domain is that there isn't a clear optimization of the stimulus that has been used in the past, which may be the reason for the change in the summation ratio. And obviously very limited information is known about the summation ratio and the advantage of binocularity in the temporal domain. So the rationale of the present study was ideally to optimize the stimulus parameter that have been varying in the past and to find what is the cause of this intersubject variation and a huge range in the summation ratio and why is it not comparable to the spatial domain. Besides that, the few other important questions that have been asked and answered in this present study is that number one, monocular and binocular modulation thresholds that were measured in the temporal domain, are they different from each other? Number two, the two optimization that have been done in the present study called as a rod enhanced and cone enhanced condition, do they differ in the flicker modulation threshold? Number three, the summation ratio, does it depend upon the target size that have been used or the location, the various eccentricity? And the last one, what could be the potential factors that could address the changes that have been noticed in the summation ratio? So in order to answer these questions, we had used the advanced vision and optometry test called as the AVOT unit. And in that AVOT, we had used the Flicker Plus program. I would briefly orient you towards the stimulus first and then the procedure that was carried over. So if you see over here, panel A shows the rod enhanced target and panel B shows the cone enhanced target. Basically, there are four flankers with a cross in the center, and the target appears with different temporal frequency, keeps flickering at 5 different locations at the center and the 4 quadrants that you see. Now, the quadrants that we mentioned is 5 degrees apart from the center, and this remains the same even for the cone enhanced condition. So this is how the flickering target was shown. Now, going to the response from the subject is made to sit 1 meter away from the monitor, and this is for rods and this is for cones that has been measured separately. The subject is also given a response box where the buttons are embossed, similar or simulating the target location that we see. So a center button and 4 peripheral buttons, they had to respond where they saw the target. So this is the adaptive staircase procedure, and the average of 6 reversals was taken as the flicker modulation threshold that was measured for the subject. So a total of 13 subjects were involved in the present study. I'm going to just give again a brief detail about the protocol that we had carried forward. Again, there are two conditions, the rods and the cones, that were tested separately. And I was talking about the optimization of the stimulus parameters. What I meant by optimization is that the stimulus parameters that you see over here, the drop down, the temporal frequency, size, chromaticity, scotopic, photopic ratio, etc., have been changed in such a way that it enhances only the cones and enhances only the rods whenever they are tested. This has been validated and published in the past by Hatti Beligal et al. In the year 2020 and 2021. Now, with this optimization and two conditions that are there, we also introduce 5 different target sizes. Now, these 5 target sizes, starting from the smallest 7 minutes of arc to the largest 60 minutes of arc, were used in the study. The thresholds were measured binocularly and monocularly, and the BSR was calculated using the dominant eye or the better eye monocular threshold divided by the binocular threshold. A little more detail about the protocol over here. Basically, we had 2 sessions. That is, we repeated each session, each of the target size twice in order to have a repeatability of the data. So the target size that we had used was 7, 15, 30, 45 and 60. And each of these target size were repeated twice. And twice, what I mean is they were measured monocularly separately for the right eye and the left eye, and binocularly each twice. So at the end, the rods had a total of 30 sessions and cones had a total of 30 sessions. Overall, approximately 8 to 9 hours for a subject to complete both the tasks. So the results that I'm going to show over here is the average results of 13 subjects who were involved, and I would be taking you over the questions that we had and the answers to the questions. So the monocular and binocular FMT change, as you can see over here, this has been plotted simulating the location of the target that we had used in the study. The x-axis over here represents the target size and y-axis represents the modulation threshold. Now, clearly, 2 critical points comes out of these graphs. One is, yes, there is a difference in the monocular and binocular responses in all the quadrants and in the center. Number 2, the systematic reduction in the FMP has been noticed as the target size increased. There is a clear reduction and significant reduction from 7 to 60 has been noted. These are the 2 important points that came out. Now, does the flicker modulation threshold change between the rods and cones was the second question that we had. Yes, there was a clear change in the flicker modulation threshold, especially if you see in the center and the quadrants, there is an overall drop in the flicker modulation threshold. Cones were a little more sensitive than the rods. So these are the results that we had found. The systematic reduction in the threshold that was consistent to Rico's law. And if you see over here, there was a clear difference between the binocular and monocular measurements. The cones were obviously more sensitive than the rods, that is, their thresholds were lower. Now, moving on to the second part of the outcome variable, that's the summation ratio. The question that we had was, what's the relationship between the summation ratios and the target size that were used? Again, if you see over here, the x-axis remains the same. The y-axis, the BSR that was measured, and the red and the black marker reports the rods and cones respectively. So the red marker that you see, that's the cones BSR, did not show any specific trend. On the other hand, rods did show a small trend. They sort of increased as the target size increased, but that was not statistically significant. And does the change in the FMT, what would be the potential factor that would cause these BSR to have no relationship with the target size? So in order to answer this systematically, what we have done is we have constructed a 2x2 contingency matrix where we had used 2 important factors. One is the interocular difference in FMT and the other one is the monocular FMT. And we had taken a cutoff mean that was there for both the variables, less than the mean and higher than the mean. In other words, the intraocular difference when it was smaller and higher, and the monocular FMT when it was smaller and higher in all these 13 subjects are plotted. So these 260 instances that were there means that these were the total number of instances that these subjects had. And out of these instances, what were the summation ratio when the IOD was large and when the IOD was small, and the same goes to the monocular FMT. So if you see over here, the first two cells that are there, n is equal to 35 and 39, where the IOD was higher, the summation ratio magnitude became low. No matter or regardless of what their FMT, monocular FMT was, when the IOD was higher, the summation ratios were poor. In the same way, in the same cohort, when you see the inhibition, the binocular inhibition, the inhibition was more when the IOD was high and it was less, the magnitude became lower when the IOD was smaller, regardless of what their monocular FMTs were. So yes, the summation estimates were higher for rods and cones in their specific location. It was obviously higher than the BSR. There was a variability in these estimates, more pronouncedly seen in the rods than the cones, and there was no reliable association when you vary the target size and measuring the summation ratio. So to conclude with the four questions again, yes, there is a change in the monocular and binocular FMTs when you change the target size, but the change was not significantly different from the locations that we tested. And the FMTs do change between the two optimized conditions. The cones were more sensitive than the rods, and the BSR were remaining independent of both target size and location. They did not follow a specific trend as we hypothesized. And the change in the BSR could be attributed to the interocular difference regardless of what the monocular FMTs or the monocular thresholds were. So a similar monocular thresholds in both the eyes would summate the flicker information better binocularly. Thank you.